Good evening, ladies and gentlemen. In this webinar, I'll be talking on ectoparasites, which are associated with the small ruminants. In general, the ectoparasitic prevalence is quite high. If you make the average of this particular table, you will find it is up to 40 to 50% is the prevalence. So every alternate animal is infested with one or the other type of ectoparasites. And many cases, in fact, represent the mixed infestation. So they are infested with the ticks also, then lice also, and sometimes a common infection with the mange also. The farmers community, they are not very serious in tackling these problems. For them, it is like a usual entity on the body. And in fact, they wouldn't know that because of these unwanted guests on the body, they are also suffering from the economic losses, depending upon the time spent on the body of the individual or the host. We can classify them into two groups as residents and visitors. As residents means, they spend extended time on the body of the infested sheep or goat, and visitors, they live away from the host and frequently visit the animals in order to derive certain nutritional benefits. So in resident group, as you can see here, there are ticks, then there are lice, different types of mites. In fact, lice and mites, they are the permanent parasites. They don't have stages which live away from the host. There are certain stages of the ticks which live away from the host, but lice and mites are permanent parasites. As far as the visitors are concerned, basically these are fleas. The fleas also stay on the body for some time, but their immature stages and even the adult stages live away from the host, and then they frequently attack the host. And that is how they transfer from one individual to other individual. And then flies, as you all know, that these are basically the visitors. For a short period of time, they visit the host in order to get certain nutritional benefits. So I have prepared a two separate chart. This is a chart for sheep. So in sheep, there are 4 different species of ticks. Haemoprotozoa is the one which is very common, followed by Hyloma and Rhipicephalus. Lice infestation is quite common in sheep with the Bovicola capre. Even on sheep also you will find this, even though the capre means a goat, and then Linognathus africanus. In mites, you have Psoroptic, sarcoptic, and Demodex. Demodex mites are less common, or it is exception rather than a rule. In case of fleas, Tinocephalidus felis is the most common flea occurring on sheep. And then Myas is the list, as you can see, flies. The list is very big. I have categorized them into 2 groups as a myiasis producing. That means they produce maggots in the different tissues of the infected host, and non-myiasis, these flies basically have a nuisance value. Okay, so this is the chart showing residents and visitors of sheep. Of course, these are unwanted residents and unwanted visitors. Then this is the chart showing the prevalence of different ectoparasites on goats. More or less, the same species are there. But as I've said, my main intention was to emphasize that they are occurring on sheep. They have been reported to occur on sheep in India and also on goat. So that is why, say about 80 to 90% of the ectoparasites are common. But when you are dealing with ectoparasites of these 2 important species of host, one should give information host-wise, and that is why I have tried here. So the lice, mites, these are same. Fleas are also. There is one reference of Tinocephalidus canis occurring on goat, but that is a solitary reference. It could be, misdiagnosis, or because in general in India, even on companion animal, the one which is widely prevalent is Tinocephalidus felis. And myiasis producing flies are also there. Barring 1 or 2 exceptions, most of the ectoparasites are common. Then let us talk about the harmful effects of these ectoparasites. And they can be categorized into 3 different groups. One, because of their presence, because of their biting tendency and a blood sucking tendency, they cause a skin injury. Then there are lesions, and because of these lesions, the animals are vulnerable for certain other infections, maybe bacterial infections, maybe other parasitic infections. Then some of these ectoparasites are responsible for causing skin diseases like mange or myiasis. Mange is caused by different types of mites, and myiasis, as you all know, caused by the larval stages of the flies, which invade different tissues. Then the allergic conditions also develop in these animals because of certain ectoparasites. All ectoparasites do not lead to allergic conditions, but certain species, I would like to name here, that is sarcoptic scabi, is responsible for causing allergic conditions. Then some of these are bloodsuckers. They are voracious bloodsuckers, and when they are present in large numbers, they are responsible for a substantial blood loss. And for example, if they are present on young ones like lambs and kids, then the blood loss is extensive for them, and then it leads to anemia. For a prolonged infection may lead to hypoproteinemia. So in general, these animals become weak, emaciated. The young ones do not grow properly. And the most important harmful effect is their ability to transmit number of diseases, their vector potential. In fact, why we are studying all these ectoparasites in rather greater detail is mainly because of their ability to transmit the different types of diseases. And these diseases, some of these diseases are really very, very deadly diseases. So to name a few, like Thalaria lestocardi, Thalaria ovis, Thalaria annulata, then Thalaria lewinsioni, Babesia ovis, Anaplasma ovis, Anaplasma marginal, Trypanosoma ivansi. Then if you look at the reflection of these harmful effects, or what is the adverse impact on the industry, as I've said, there is a loss of production, maybe a milk production or a meat production. And in case of meat production, particularly in sheep, there are myiasis producing flies. There are around 6 to 7 myiasis producing flies occurring in India in different seasons. And when the myiasis is extensive, then even a subcutaneous tissues are also involved. And in such cases, there is extensive carcass trimming, and even the meat is also downgraded, and that is how the farmers have to sustain the economic loss. Needless to say, they are present on the body of the animals, and there are skin lesions, and as a result of this, there is loss to the quality of the hide, or a quality of the skin becomes very rough. So the leather industry also suffers because of these ectoparasites. To quote the example, I can quote myiasis and mange, where the skin is damaged extensively and the hide quality goes down. Then loss of wool production is also one of the important trait. And then in general, because of all these ectoparasites, the reproductive potential of the animals also goes down. So if the young ones are affected, like lambs and kids, then they don't grow properly. They don't attain puberty at the right time. And even, their reproductive cycle is also affected, and there is economic loss to the farmers. So these are the adverse impacts of the ectoparasitic infestation. So let me talk about the enemy number one, and that is the tick infestation. We have seen these are, so as to say, the residents on the body, and they are present in large numbers. Haemaphysalis is the common species, Haemaphysalis cuchensis, Haemaphysalis bispinosa, then Rhipicephalus haemophysalitis, and I'm reading the list occurring on sheep. Rhipicephalus sanguinus, there are few cases. Hyloma species, Hyloma marginata misaki and Hyloma anatolica. On goats, you have same species, Haemaphysalis, Rhipicephalus, and Hyloma. I could see Hyloma marginata misaki. I could not find the reference of Hyloma anatolicum. on goats. Then the Haemoprotozoan infections, which are transmitted by different vectors. Basically, it takes Thalaria lewinsiani, Thalaria ovis, then Thalaria lestocardi. This is a new name. Lestocardi is a new name for Thalaria hirsi, and it is the most pathogenic species of Thalaria. Then Trypanosoma evanzi, obviously transmitted by different blood-sucking flies like Tabanus and Stomoxys. Babesia ovis is also very much prevalent in India in sheep. Anaplasma ovis and Anaplasma marginal. Anaplasma ovis and Anaplasma marginal, in addition to ticks, are also transmitted by blood-sucking flies. On goats, you will find that the species are more or less same. So these are species occurring in goats also, and they are responsible for creating a disease, and even in a subclinical state also, they are responsible for a substantial economic losses. And these diseases are transmitted by ticks. There are two types of transmission, as a transstadial transmission and transovarian transmission. In transstadial transmission, the nymphal stages transmit the infections which they have acquired during the larval stages, and adult stages transmit the infections which they have acquired during the nymphal stages. And transovarian means the organism, that means the different types of protozoa, bacteria, viruses, they appear in the eggs of the ticks, and the larvae and nymphal stages of the next generation are involved in the transmission. So these are vectors which are involved in the transmission of infections from the infected animal to clean animal. Prevalence of these Haemoprotozoan infection is quite high in certain parts of India. When you want to control the ticks, one must know about the biology of the ticks. I am going to touch only those points which are relevant as far as the field conditions are concerned. As we all know that the ticks are ubiquitously distributed throughout India, and to some extent they show seasonal dynamics or a seasonal preponderance. Generally, the tick population is more during a hot and humid climate. So what is the biology of the ticks? The ticks have 4 distinct stages in their life cycle. The eggs are there, which are laid on the ground. Then the larval stages, these larvae have 3 pairs of legs, or a hexapod larvae. Then the larvae are converted into the nymphal stages, which have 4 pairs of legs, but there are no genital organs. And then nymphal stages are converted to the adults. And these adults also have 4 pairs of legs, but they have genital organs very well developed. And each stage in the life cycle, that means larval stages, nymphal stages, and adults, male and female, they are hematophagous in the sense they attach themselves to the body of the animal and they suck blood with one go, and that process is known as engorgement. When the process of sucking blood from the host is known as engorgement. So the larvae engorge, nymphal stages also engorge, and the adults also engorge. There are 3 basic types of ticks depending upon the life cycle stages, like 1 host tick, 2 host tick, and 3 host tick, depending on where the molds are occurring. Okay, there are 2 molds in the life cycle. As you can see here on the screen, larvae to nymph is the first mold, and then nymph to adult is the second. So there are 2 molds, larvae to nymph and nymph to adults. And depending upon the location of these 2 molds, the ticks can be classified into 3 types as 1 host tick, 2 host tick, and 3 host tick. In 1 host tick, both the molds occur on the body of the individual. Now, we don't have an example from sheep and goat, but in cattle, Buphyllus microplus or Rifficypalus microplus is a 1 host tick. So, where both the molds from larvae to nymph and nymph to adult occur on the body of the same individual. So, 1 host tick does not mean that only one animal is required. It means that the animal is required once for completion of life cycle. Once the larvae attach themselves to the body of the individual, then the rest of the stages, nymphal and adult stages, are formed on the body of the same individual because both the molds occur on the body of the same individual. But in case of sheep and goats, we have example of a 2 host and 3 host tick. Basically, most of the ticks occurring on sheep and goat in India are a three-host tick. So in case of two-host tick, the first mold, that is from larva to nymph, occurs on the body of the animal, while the second mold, that is nymph to adult, occurs on the ground in the cracks and crevices. And that is why it is called as a two-host tick. That means it requires host twice for completion of life cycle. First time as during the larval stages for attachment, and second time when nymphal stages mold to adults on the ground, then the adults also require the host. So the host is required twice, and that is why the ticks are called as two-host tick. And then three-host tick. As I've said, on small ruminants, most of the ticks, what we have seen, Haemoprotozoa, Rhipicephalus, Hyaloma species, they are basically three-host ticks. So if you look at the life cycle, three-host tick means there are two molds and both the molds occur on the ground, okay, in the cracks and crevices. So this is the pattern. The female, after blood-sucking activity, they drop down, and then on the ground, they deposit large number of eggs. It has been estimated that one Haemoprotozoa female lays around 4,000 eggs in the cracks and crevices. Then the larval stages, they attach to the body of the host, maybe sheep or a goat, and then after that they suck blood. So attachment followed by the engorgement, and after engorgement, after the blood sucking activity, the larval stages also drop down. And in the cracks and crevices on the ground, the larvae mold to nymphal stages. So this is the first mold occurring on the ground. Then the nymphal stages, once they are formed in the cracks and crevices, they then attach to the body of the same individual or a different individual. And after attachment, these nymphal stages also engorge. They also suck blood. And after engorgement, the nymphal stages drop down, and on the ground, the second mold takes place. So the first mold from larvae to nymph also takes place on the ground, and the second mold from nymphal stages to adult stages also occurs on the ground. And then the adult stages, once they emerge out from the nymphal stages, then these adult stages also attach themselves to the body of the host, same host or a different host or individual. Then they also engorge, then the mating takes place on the body, and then the female tick drops down and lay eggs in the cracks and crevices. So on your screen, you will see orange boxes. Orange boxes indicate the development of the body or on the ground in the cracks and crevices. And here you will find the first mold occurring on the ground as well as the second mold is occurring on the ground. So in this case, this tick is called as a three-host tick because it requires the host three times. Even though in that particular premises there is only one animal, then also the life cycle can be completed because three-host tick means the host is required three times. Now, on this particular plate, you will also see certain figures. Now, this is, I would like to just emphasize the magnitude of multiplication within a span of say 1 to 1.5 months, how the tick population increases. As I've said, that one female tick lays around 4,000 eggs. Now, by considering some adverse climatic conditions or even by considering the law of averages, if you say that out of those 4,000 larvae which hatch out from the eggs, only 2,000 larvae, they find their host or they succeed in finding the host, then the larval stages develop, engorge, they drop down, and the larvae mold to nymphal stages. And then out of 2,000, say 1,000 nymphal stages, by law of averages, 1,000 nymphal stages, if they find the host, then they suck blood, they drop down, and then they are converted into male and female. So by law of averages, again, from 1,000 to 500, and in that 500, say 250 are males and 250 are females. And then each female at the end of the cycle lays around 4,000 eggs. So if you do simple math, that there are 250 females in F1 generation, each lays of 4,000 eggs. So within a span of 1 to 1.5 months, because this cycle requires around 1.5 months to get completed. So within a span of 1 to 1.5 months, 10 lakhs of larvae of next generations are formed. And this is out of only one female. So now you can imagine the magnitude of tick infestation and how rapidly this tick infestation increases. So that is my main intention, to emphasize the magnitude of this tick infestation. So now when you are dealing with the tick infestation, you have number of options available. Nowadays, people are talking about integrated tick management or integrated pest management. No doubt that we should tackle these problems or a problem of ticks, but we should tackle in such a way that it will lead to a win-win situation. That means the ticks are eliminated, but at the same time, the environment is not affected. There is no biodegradation. There is no adverse impact on the animals and even not on the environment. Nowadays, we talk a lot about the One Health program or a One Health concept. What is One Health? One Health is, it is the health of the animals, health of the human beings, and also health of the environment. So one should consider the health of the environment also. So your approach should be in line with integrated pest management. So what is that approach? What options you have? In the first place, first is mechanical or the physical steps. In the sense, you can pick up the female ticks and then destroy them. As you know that one female tick can lead to 10 lakhs of larvae of the next generation. So even, if you take out the 10 or 15 female ticks, then you are going to reduce the environmental contamination to a very great extent. It might appear a little foolish that, taking out the female ticks, but if you want to prevent the subsequent infestation or even minimize the subsequent infestation, it is worth attempting. Then destroying the stages which are hiding in the cracks and crevices. Because as I've said, that in the life cycle, the larval stages, nymphal stages, adults, they are hiding in the cracks and crevices. So you can use a scalding method or a burning method, and then on the pasture land or on the ground, if you do the plowing operation, then the soil is turned upside down, and then the stages which are hiding deep inside the cracks and crevices are exposed to hot sun, or even you can use certain acaricide preparations. There are certain herbal preparations available, those can be used. Then second aspect, which is commonly practiced by the field veterinarians and even the animal owners or the farmers, and that is the use of chemotherapy. There are a number of compounds which are available, like inorganic compounds nowadays. These are not available, but we started with that. Then organochlorines, organophosphorus compounds, carbamates, pyrethroids, ivermectins, formamidins, and there are at least 40 to 50 herbal preparations, formulations, which are commercially available. So those can be used. Then as far as the biological control is concerned, you can use different types of bacteria, fungi, and nematodes. Of course, the commercial products are available to a very, very limited extent. Now, for cattle, the vaccines are available, but the same vaccines are not useful for sheep and goat, because the species that are occurring on sheep and goat are altogether different. The vaccines which are available for cattle are prepared from Boophilus microplus, while this kind of tick is not very commonly encountered, or it is very rarely encountered on sheep and goat. So the vaccines which are designed for cattle are not useful here. And biological control, as I've said, you can use a different biological enemies, and those biological enemies can take care of the ticks and their immature stages. So that is the approach of integrated pest management. You should try to use the methods which are very eco-friendly, which will not disturb the ecological balance. That is the main focus. So you have to integrate different means or different methods for the effective management without having an adverse impact on the animal population, human population associated with the activity, and also the environment. We should begin the tick control step by step. The step one is a physical or a mechanical. As I've said, the manual removal of big fat ticks will reduce the environmental contamination to a great extent, because when you destroy these ticks, the biotic potential is reduced to great extent. In fact, when the tick is destroyed, the biotic potential of that particular tick is zero. Then burning and scalding. This is also very effective method, and I think I've got a picture of this, so I will explain in more detail on that particular plate. So when you do this, there is the environmental contamination comes down to a great extent. And when environmental contamination comes down, there is less chances of the animals getting a new infection, because the source of infection is curtailed to great extent. So first is a mechanical means or a physical means, where all the physical activities are performed to control the ticks. Number two is the herbal preparations or biopesticides that can be used. Now, some of these biopesticides are commercially available, or the herbal preparations are commercially available. As you all know, in the market, you get the neem oil, currants oil, eucalyptus oil, and there are 7-8 different types of oils are available in the market, and at a very reasonable rate. So you can use this particular product, and then I'm going to tell you how it is to be used. So it is easily available and it is eco-friendly also. Then there are certain biological agents which are commercially available. In fact, these agents have been developed as the agricultural pesticides. So the same can be used for control of ticks. Okay, there are bacteria. This bacteria, Bacillus thuringiensis, this powder is available. The spores are available, and the fungus, Metarhizium anisopoli, the fungus is also commercially available. So first is a mechanical means. Second is the herbal preparation or biopesticides you can use, and try to restrict the infestation of the ticks and other ectoparasites. And if you are not in a position, unfortunately, to control to a desired level, then you can think of using some chemicals or a chemical acaricides. Okay, so that can be used only if required, and that too in emergencies. So this is what I was talking about, a physical means or a mechanical means. As you can see here in this particular picture, this is the tick, and the eggs are being laid here in the cracks and crevices. These are some ticks and the immature stages. So what you can do, on a stick, you can just put a cloth and then little kerosene, and you can prepare this kind of a mashal, and then with this you can do the scalding. In our Parbhani Veterinary College, one of my colleagues, Dr. Narlatkar, has done a wonderful work. What he did not use anything on the body of the animal, but for 1 year he used only mechanical methods, deticking and scalding and burning of the cracks and crevices or the walls, so as to minimize the environmental contamination. And the results were also wonderful. About 80 to 82% of the population was reduced, or the population was reduced to 80 to 82%. So it is a great achievement by using just physical means. We are not using any kind of chemicals, but at the same time, in a span of 1 year, the population came down by 82%. So how these products which are easily available even in rural areas can be used? So herbal formulations, I've got 2 formulations with me. One is this currant oil or a neem oil or eucalyptus oil. This is available in the market. What you can do, you can take 15 ml of this oil and then mix about 2 grams of liquid soap in it, and then 1 liter of water. So 1 liter of water, 2 grams of soap, and in this 15 ml of either neem oil, currant oil, or eucalyptus oil. And then mix it, keep stirring continuously because you are preparing the emulsion. The oil doesn't get dissolved or mixed in the water. So the soap is like a saponin. It forms a emulsion, and then keep on doing it. And this mixture is allowed to stand for 6 hours. And then this particular mixture can be used for spraying on the animal body. It is absolutely safe, and you can spray this particular mixture at the rate of about 0.5 to 0.75 liter per adult sheep or goat, weighing around say 70 to 80 kg or even up to 100 kg. So you have to use a spray and just do the wetting. Complete the body should be wet, and about 0.5 to 0.75 liter is required for one animal, and that you can do it on a monthly basis. And you can also use the same formulation or a preparation for treatment of premises at the rate of 30 ml of this formulation per square meter, and that too on a monthly basis. So this is also yielding a fantastic result. So let me go to the second formulation, and here it is the neem fruits. Particularly in summer, you see the neem trees with these yellow colored fruits, and these fruits are, on one tree there are thousands and lakhs of fruits are there. So collect all these yellow colored fruits, which are called as the neem fruits, generally available during summer. Having the scientific name of this particular plant is Azedrica indica, and there are 108 active ingredients in these fruits. So take 5 kg of these fruits, grind them finely, okay, make it like a paste or a powder, and soak it overnight in the water. Then filter it through the muslin cloth or any ordinary cloth and add 200 grams of liquid soap, and then add water to make the total volume 100 liters. Okay, so this can be prepared for a big farm having 100 to 150 animals. And even if you want to do it for a 20 to 25 animals, you can reduce the quantity in that proportion. And then the same solution can be used for spraying on the animal body at the same rate, 0.5 to 0.75 ml per animal. And also on the infested premises, in the cracks and crevices or in the animal houses, once in a month, and with the same rate of application, that is 30 ml per square meter. So two formulations we have seen. One is a commercially available neem or a currants or a eucalyptus oil that can be mixed with the liquid soap and tap water. And second is these fruits can be used. And I'm pretty sure in rural areas, these are available. And in fact, you can store these fruits in a powder form. You can dry it and store it in a powder form. And then subsequently, you can use it till the next summer. So a very good herbal preparation having having a beautiful effect on the ticks and can be used without having any adverse impact on the environment. Then let me talk about the biological control by using natural enemies. As I've quoted two examples, one is a Bacillus thuringiensis, which is a bacterial spore powder, which is available commercially. The details are given here. This is available in India and commercially. So what you have to do is that this is the trade name, Dipel 8L is the trade name, which contains the mixture of toxin and bacteria, about 17,600 international units per milligram. And that solution contains 3.5% weight by weight basis. So you can use this for controlling the ticks, larval stages, nymphal stages, eggs, but not on the body of the animal. This is to be exclusively used in the premises. You cannot sprinkle this, spray this on the body, mind it well. The previous two formulations we have seen that you can sprinkle on the body or spray on the body, but not this. Okay. So in addition to the sidal effect, this particular product also reduces the reproductive potential of the female ticks. In the sense, the number of eggs laid by the female tick is also less. Those eggs, majority of them fail to hatch, and that is how the reproductive potential of the tick is brought down. So a wonderful product, and this is commercially available, and you can try using it without using those dangerous acaricides. Then the fungus, which is also commercially available. The name of the fungus is Metarhizium anisopyle, which is commercially available in the powder form like spores. It is available in MPKV, that is Agriculture University at Rahuri, and 1 kg packet is available for 100 rupees, but this packet is, you can use it for a number of times. What do you have to do? Let me talk about the fungus first. So this is a commercial packet available in the market. This is the powder which is kept in a bowl. So you can see the appearance of the powder, and this is the microscopic appearance of the fungus. These are the spores, and then hyphae, and then septas. So this is the fungus. Even if you take a little bit of powder, mix it in the water and pip in the microscope, you will see this particular structure. How it is to be used? 5 grams of this fungal powder is taken in 1 liter of water. You can add 5 grams of jaggery. Jaggery is a kind of nutrition for the fungus to grow. Okay, or a good, what we call. Or if good is not available, you can even add 5 ml of milk in this, mix it well, and then apply it in the premises. You can't use it on the body. Okay, the scientists are working, in the laboratories to check its efficacy, and more than that, safety to the animals when it is sprayed on the body. But at present, we are recommending that you use it for a treatment of premises. And in the premises, particularly for 3 host tick and 2 host tick, number of stages are hiding in the cracks and crevices, and those stages can be targeted with this particular type of formulation. And the rate of application is same, 30 ml per square meter. So you can now imagine 1 liter of solution, 5 grams are required. The packet is available 1 kg for 100 rupees. So you can use number of times this particular product, and with thousands of square meter can be covered with 1 kg of this particular powder. So it's quite economical for the farmers also. This is the microscopic picture, how it happens. These are the normal eggs. So in the laboratory, when the eggs were treated with this fungal spore, those eggs, they die. And then you can see here the dead eggs and the larval stages. When they are in a live condition, they are creamy in color, and even the larval stages are whitish in color. So you can see here, you can appreciate the death of the eggs and the larval stages because of the fungus. So this fungus, traps the eggs and the larval and nymphal stages from all the sides, and then ultimately cause death of these stages. Now, one more method, what you can try. I did not find a very, what should I say, a scientific reference for this, but there are a number of indirect references stating that you can use a chicken or a fowl, desi birds, to control the tick population, because these birds are basically insectivorous, and they keep on eating, small soil creatures like beetles, insects, earthworms, ticks, and mites. These are not mange producing mites, but a soil mites, oribatid mites. So they go on picking. If you let loose these birds on the ground, you will find they are picking up something from the soil, and they are basically insectivorous. So I could get only one reference, that when the hungry birds are let loose in a shed for 30 minutes, they can wipe out or they can eat about 70 to 80% stages of the ticks hiding at different places or those which are at the surface. So you can also use a chicken for controlling these stages. These are all small, tips which can be implemented, and by doing so, we are delaying the use of dangerous acaricides. And the third step, unfortunately, if the first two steps are not adequate to control the tick population, or when the farm is very, very heavily infested and a quick control is required, then you are left with no other alternative but to use different types of acaricides. So in that case also, you should adopt to one particular policy or a strategy. What you can do, you can start, first applying these chemicals. The first two steps do not work, then you can go. And then treatment of the premises. Start with only treatment of the premises first. See how it is responding, And if the premises treatment is also not giving you the desired effect, then you can go for application on the body. My personal recommendation is that you use pyrethroids more frequently for treatment of these ectoparasites and save ivermectin or amitraz for future use or for future emergencies. Because as you all know, ivermectin also has a adverse effect on the intestinal worms or gastrointestinal worms, and if it is used indiscriminately, then there are chances. So you have to use these chemicals very sparingly or very judiciously. My recommendation is that you use pyrethroids. So by adopting all these things, you are protecting your nature. But when you are using chemotherapy indiscriminately, because these insecticides or acaricides are available across the counter, no prescription is required. You can just buy it and use it. And the farmers are also using it. So what are the adverse impact of use of these acaricides indiscriminately? There is a possibility of toxicity in animals and human beings. Then there is a biodegradation. That means the other useful fauna, soil creatures, or fauna present in the environment is also destroyed. So there is a biodegradation, the ecological balance is disturbed. Then the residues are formed in the natural resources, in the water and in the soil and in the plants. The residues are also formed in the body. Okay, so the residues in the meat, milk, even the residues are formed in the skin and in the wool. Then if you use it indiscriminately, there is a possibility of a development of resistance to these different ectoparasiticides. And a long-term use may even lead to carcinogenicity or teratogenicity. So these are all adverse effects. And that is why the emphasis should be, to control these ectoparasitic infestation without using, as far as possible, or minimally using these chemical acaricides. So that should be the approach for tackling ectoparasitic infestation. Now, what happens in case there is a toxicity? What should be the approach to tackle the cases of toxicity or poisoning? Number one is that you should know the antidotes. For example, for organophosphorus compounds and for carbamates, the antidote is atropine sulfate. So you can use atropine sulfate. Then picrotoxin is the antidote for ivermectin, different types of ivermectin. Amitraz, my recommendation should not be used as far as possible, but if you use it and if you come across a toxicity, then yohimbine is the antidote. Of course, on the label, you will get the instruction regarding how to use this in case of toxicity. While for organochlorines and pyrethroids, there are no specific antidotes available. So you have to treat or deal these cases on the basis of symptoms. Then the second aspect is decontamination. The first, you should decontaminate the surroundings, okay? And a supportive care must be provided to these animals. So for example, if the toxicity is a result of the topical application, then you can think of washing the animals and removing these chemicals. Then gastric lavage, or you can use activated charcoal for binding with these chemicals and reduce the systemic signs. Then the animals should get fresh air. There should be proper ventilation. And then you have to support the animal symptomatically. Whatever symptoms are there, you should use the drug to counter those symptoms. Mostly these are neurotoxins, and the nervous signs are seen. In majority of these cases, there are convulsions or hyperesthesia. So anticonvulsants can be used in addition to the specific antidotes wherever possible. Then precautions one has to take. Number one, whenever you are using ectoparasiticide, go through the literature first. Read the label. In some of the containers, there is a leaflet inside. Go through this leaflet very, very carefully. Be prepared. You can't, start searching things, after you come across toxicity cases. So be prepared for it. Then special care should be taken as far as the young ones are concerned and the pregnant animals are concerned. And then whatever these containers or the boxes are there, or the empty containers are there, they should be, disposed of very, very scientifically. They should not be easily accessible. So that is also one of the possibility of the poisoning or toxicity. So that should be the approach towards dealing the toxicity cases or poisoning cases. Now, this is about all tick infestation. And by explaining the tick infestation first, I have almost 60 to 70% covered the use of ectoparasiticide. Now we will go one by one. First we will see lice infestation, then flea infestation, then mite infestation, and fly infestation. So as you can see here, this is a lice infestation. We can see either these are the adult stages or the nymphal or the eggs, which are called as the nits. Here you can see there are adult lice on the body. So let me deal with the lice infestation. As I've said, lice are permanent parasites. Okay, all the stages are present on the body of the same individual: eggs, larvae, nymphal stages, and adults on the same individual. And then the infestation is transmitted through sheer contact. Occasionally, when the farm appliances are also contaminated, because these lice can survive away from the animal body for about 1 to 2 weeks. So when the farm appliances are also contaminated with the life cycle stages of the lice, then these appliances are also responsible for the transmission of lice infestation. So that is how lice infestation is transmitted. There are basically two types of lice. One is a sucking louse and second is a biting louse. The sucking louse has a pointed head. If you look at the head, it is conical. While biting louse, the head is broad at the anterior end, or it is rounded. If you look at the mouth parts of these lice, you will find the mouth parts of sucking lice are piercing and sucking type, like mosquitoes. And that is why they are on liquid diet. So they suck superficial lymph, or occasionally they suck blood, because the mouth parts are piercing and sucking type. In case of biting louse, the mouth parts are biting type or a chewing type. And these lice, they feed on the solid material like dandruff, dead epithelial cells, and other structures on the body. So that is how they swallow these things. Then usually sucking lice are present on the head and on the legs. Lice are not only rigidly host specific, but to some extent they are also region specific. And that is why if you go through the literature on lice, you will find different common names like head louse, foot louse, different names are given. Okay, body louse. So that indicates the region where they are frequently found. Now, in case of a biting lice, they are present on entire body. The species Linognathus tenopsis is a sucking louse, Linognathus africanus. Then these are present on goats, on sheep, Linognathus pedalis, which is called as a foot louse. I don't say these two lice do not occur on sheep, but whatever literature which was available to me, in that literature, I did not find any reference stating the occurrence of these two species on sheep. Then in biting lice group, on goats, there is bovicola, the new name for Damalinia capre, and in sheep also the same louse can occur, that is a bovicola or Damalinia. On sheep it is also known as bovicola ovis, depending upon the host on which they are occurring. Now, clinical signs, when the infestation is very mild, you will find that the body coat is rough, but as the infestation rate goes on increasing or piling up, because they are permanent parasites, so the infestation goes on increasing slowly, you will find they are scratching the affected portion of body, or in general scratching is there, rubbing, with the help of their hind hooves and horns, they keep scratching. They rub their body against the wall or any other object. And sometimes, they try to bite the portion of the body and pull out the hair. So these are the clinical signs of lice infestation. And in case of wool, you will find the wool is not the normal wool. It is disturbed and discolored. We call it as a disturbed or discolored fleece. So these are the clinical signs of lice infestation. And one, if you examine the body coat very carefully, these are macroscopic. You can locate lice on the body. So the adverse impact, as I've already covered, there is a adverse impact on the skin or on the hide. So the leather industry is adversely affected. Wool industry is also adversely affected, and there are production losses also. See, when it has been proved, when these infestations were treated, the production also increases, because when there is an increase in the quality of the life, then the performance is also better. Regarding control of lice infestation, with or without shearing, because it is particularly in sheep, it has been mentioned that if you do it after shearing, the efficacy of these different preparation is quite good. So topical applications can be used, like you can use the products available in a powder form for dusting, then dipping and spraying. Different formulations are available. Of course, we are going to see what are the different formulations that are available. Then regarding management, one has to examine the body coat carefully, avoid overcrowding because the infection is transmitted through contact, cleanliness of the farm appliances, and if the new animals are purchased, then these animals should not be allowed to mingle with the other animals immediately. You should examine these animals, observe these animals for number of infections and infestations, including lice infestation. So that should be, the strategy for control of lice infestation. And in fact, any ectoparasiticide, whenever you come across ectoparasitic infestation, with the first instance, you have to tackle it. Before it goes out of proportion, you should tackle it. Then flea infestation is not very common on sheep and goat, but there are reports, and the flea which is reported on sheep and goat in India is Tinosyphilidus felis, var orientalis. There are 4 different types, but the one which is widely prevalent in India is Tinosyphilidus felis, var orientalis. There is these fleas do not cause a specific infestation. That is the contrasting feature when you compare with lice. Lice are host specific, while fleas are not rigidly host specific. So they can thrive on different hosts. Even they can climb on humans. Then the flea infestation generally on sheep and goat do not last long. So, and the infestations are mostly uneventful. The number of fleas are also less, and these fleas are vulnerable or susceptible to pyrethroids. And as I've said, for tick infestation or a lice infestation, pyrethroids should be your first choice, so that you are sparing the other good compounds which can be used for tackling mange, or if there are cases of resistance. So what are the basic differences between lice and fleas? Lice is also called as lice infestation is also called as a pediculosis. So lice, as you can see here in this picture or a photograph, that the body is dorsoventrally flattened. While in case of flea, the body is laterally compressed. And these fleas, because the body is laterally compressed, they can move very swiftly on the body. While lice, the legs are very, very short and stumpy. As you can see here, the legs are very short and stumpy. As against this, here you can see the legs are very long. And these, with the help of these long legs, they keep jumping or move by leaping action or a jumping action. While lice, because the legs are very short, their movements are sluggish. I don't know whether you have come across this phrase or not. Generally, if any individual is very lazy and lethargic, we call it you lousy, so lice. So louse is basically the movements are very sluggish. Okay, you can easily catch lice, but you can't easily catch fleas. The moment you try to catch the flea, it disappears. That means it jumps from there because they are very active. Then in case of lice, all the life cycle stages are parasitic, and you will find on the body of the same individual all these different types of stages. Okay, in case of fleas, only adult stages are parasitic. Okay, and when you see 5% or 5 fleas on the body, take it for granted that 95% of the immature stages are present in the surroundings or in the premises. So only adults are parasitic. These are permanent ectoparasites, lice. As we say, they are residents, while these are visitors. They are temporary, frequently drop off and then climb on. Then, as far as the infestation transmission is concerned, it is through contact. The animals also rub their body against each other, and that is how the infection is transmitted. If there is overcrowding, then the infestation is transmitted. And the farm appliances, if they're not cleaned properly, then the infestation is transmitted. In this case, through contact also, but through also contaminated surroundings. As I've said, 95% of the stages, which are immature stages, are present in the surroundings. Now, lice, even the sucking louse, it feeds on a superficial limb. Occasionally, it feeds on blood, while fleas are blood suckers. So they suck blood. Lice, as I've said on a couple of occasions, they are not only host specific, but region specific. While fleas, they have a very flexible host specificity. So you can't even think about the region. So these are the basic differences between lice and fleas. Now let me come to mange. I have categorized them into 3 types: burrowing mites, then non-burrowing mites, and follicular mites. Now, follicular mites, for the time being, you can ignore it because there are very few cases reported of demodicosis in sheep and goats. While these 2 cases have been reported, burrowing mites, the best example of this occurring on sheep and goat is Sarcoptis cabi, and non-burrowing mites, Psoroptis ovis, particularly in sheep, and Coreoptis bovis. There are few references on goats. So burrowing mites, non-burrowing mites, and follicular mites. Now, mange, as you all know, that mange is caused by different types of mange-producing mites. And they are also like lice. They are permanent ectoparasite. They are permanent residents, unwanted residents. And there, the infection is passed on from the one animal to other animal through contact or through farm appliances. So as far as transmission of infestation is concerned, in case of lice and mites or a mange producing mite, the mode of transmission is same. So burrowing mites is a sarcoptic scabi responsible for causing scabies or a sarcoptic mange, while non-burrowing mites, the Psoroptic mange. If you see compare their features, I will enumerate certain contrasting features. Sarcoptes, as you all know, burrowing mites. Psoroptes, they are superficial, they are non-burrowing mites. Now, sarcoptic mites, they affect the less hairy portion of the body. So you will find it, these mites or the lesions on the head and feet to begin with. While in case of Psoroptis, the lesions are formed on the entire body. Then sarcoptic mites are a burrowing mites. So these mites are responsible for hyperkeratosis. They are responsible for acanthosis, that is a melanin deposition. And that is why this mange is also known as a black mange. So there is a thickening of the skin because of the hyperkeratinization. While in case of Psoroptis, because these mites do not penetrate to the deeper layers of the skin and migrate superficially, then these mites are responsible for causing a thick scab, okay, which can be easily peeled off. So the thick scabs are formed, while here there is a thickening of the skin. Then, as I've said in the introduction, that these mites are responsible for a kind of allergic reaction, that is a hypersensitivity reaction. And in this case, there is no hypersensitivity reaction. So maybe these are burrowing mites. While burrowing, they lay eggs. There are certain excretory and secretory biochemicals which make the host sensitized in case of Sarcoptes, and that leads to hypersensitivity. And as you all know, that whenever there is a hypersensitivity, it leads to intense pruritus or itching. As compared to that, in Psoroptic mange, the itching or the Psoroptic manges are less pruritic. The itching is less as compared to Sarcoptic. If you sometimes in a prolonged cases, if you collect the blood sample, in case of Sarcoptic mange, you will find eosinophilia, while you will not find eosinophilia in case of Psoroptic mange. Because sarcoptic mange sometimes, is difficult to diagnose by skin scraping examination, because the lesions are formed in the areas where there are no mites. Mind it well, this is a hypersensitivity reaction. So you scrape the portion of the skin where lesions are there. But because there is a hypersensitivity reaction, it may be produced on the areas where the mite population is very less, negligible or nil. So if you scrape this area, you may not get the mites and their immature stages in the skin scraping. While in this case, the skin scraping examination is a very sensitive method, because the lesions are formed where the mites are present. So if you scrape it, you will find the mites. Here in this case, 25 to 40% of the cases will reveal mites. So negative result in this case does not mean negative. The case is negative for sarcoptic mange. You have to use your all clinical judgment and go ahead with the treatment. Sarcoptic mange is a very, very contagious mange. You will find if there are 5-6 animals in a shed, so you will find all the 4 to 5 animals having this kind of mange. In case of sarcoptic mites, when you are using acaricides, then the topical applications are less effective, because topical applications or the chemicals do not reach up to the mites, because these are burrowing mites. The chemicals remain on the surface. The same thing happens with demodectic mange also. The mites are present in the follicles. So the chemicals do not reach up to the mites, and that is why they are less efficacious. While in case of Psoroptis, because they are superficial, if you apply the chemicals in the form of a topical application, then the efficacy is quite good. So these are some of the comparative features of burrowing and non-burrowing mites, or sarcoptic mange and Psoroptic mange. So this is the sarcoptic mite, and this is the histopathology where I want to emphasize the burrowing nature of the mite. So tunnel is produced, and the mites are penetrating into deeper and deeper areas. These objects are nothing but the mites, and they go on depositing eggs. While in this case, as you can see here, the mites are superficial. Mites, do not penetrate into the deeper layers. And these are Psoroptic mites. You can see this is a female mite because you can see egg inside. So this picture, I got it when I was going through, Google search, showing the different regions affected by these different types of ectoparasites. So the different colors codes and the color scheme is used. Modex and sarcoptes, they are basically on the head region, and then the lice, bovicola, which are biting lice, they are present all over the body. And foot mange is like choreoptic mange. So these are some of the pictures showing the lesions of sarcoptic mange in goat. You can see here keratinization and to some extent acanthosis. Here the generalized infection of sarcoptic mange in goats. These are pictures of Psoroptic mange, and I want you to concentrate here where you can see a thick scab, and this particular portion can be easily peeled off because the mites are superficial, and you will find on the entire body. In case of sarcoptic mange, it begins with the head region, but in this case, it is very common in sheep, and it is called as a sheep scab, and the body portion is affected. As you can see here, as the wool is lost, they keep invading the portion of the skin which is normal. So that is how, and the scabs are left behind, and then the scabs can be removed very easily. Then Coreoptis bovis is not very common in India, but I've got few references. As far as the life cycle pattern is concerned, it is similar, precisely similar to Psoroptis. So they are also superficial. The only thing is that their main region is on the legs, and that is why it is also called as the itchy leg. So basically at the region of two joints, fetlock and pastern joint, and occasionally on the head also. When I went through the literature for last 8 to 10 years, I could see one or two references, and that too on goats. I did not find a single reference on sheep. Okay, so maybe more likely to occur in goats rather than sheep. And because they are superficial, whether it is a Psoroptic mange or a Coreoptic mange, these mange, the mites are easily can be tackled, targeted, and the infection is curable. It is rather tedious to cure sarcoptic mange because of the burrowing nature of the mites. So the management part is concerned, it is similar to Psoroptic mange. So Psoroptic mange and Coreoptic mange can be tackled easily, can be cured. But sarcoptic mange, you require frequent monitoring of the case and the frequent application of the acaricides. Now coming to the chemical acaricides. I have followed the same trend which I did for anthelmintics, that these are the chemical groups. Organochlorines, you don't get it organochlorines, but still the organochlorine formulations are available in the form of a cream. BHC cream is also available. Organophosphates, then carbamates. Carbaryl is a powder which is used for dusting. If you are using organophosphorus compound, this should be the concentration for the use on the body, and in the premises, you should have a double concentration. Now I am talking about all kinds of ectoparasitic infestation: ticks, lice, fleas, and also mites. Then, as I've said, pyrethroids, they should be used because the distinct advantage of pyrethroid is that it is a water soluble, so the solution can be prepared, and then the pyrethroids have a knockdown effect. Like in boxing matches, there's a knockdown effect. So as soon as the arthropods or the ectoparasites come in contact with the different types of pyrethroids, they die immediately, because basically pyrethroids are the contact poisons, and these are the concentrations you should use it on the body. Then one more pyrethroid is a flumethrin, which is available as a pour-on formulation. Even permethrin, pour-on formulations are available, and the formulations which are now available in the market, you can use it at the rate of say 1 ml per 10 kg body weight, and you have to just pour it along the back line. It gets absorbed, and then it exerts the acaricidal or insecticidal effect. Propugzar is also quite popular, and it is available in the powder form. So whenever the compounds are available in the powder form, you can use it for dusting. Then ivermectins like ivermectin, doramectin, moxidectin is also available in a pour-on form. So that can be used. Formamidins like amitraz should be used sparingly. Then phenylpyrazole, fipronil, it is pretty expensive, so the farmers cannot afford. Then salicylate, which is basically anthelmintic, endoectocide. So closantel can also be used at the rate of 10 milligram per kg. Or I have come across one or two references using closantel dip, like 6 ml per liter of water, but it is pretty expensive if you use it at that particular rate. So what are the methods of insecticide or acaricide applications? You have a dusting, then dipping, spraying, and a systemic use. In dusting, when it is available in the powder form, you can use it. You have to use a brush, okay, so that it is evenly spread on the body. So there is an ease of application. It is less tedious, but you may not achieve a uniform application. So some pockets may remain untreated and the efficacy may go down. But at the same time, there are less chances of toxicity. So I would say if the infestation is very light, if you have spotted it for the first time, you can go for dusting and try to control it by using different types of powders like carbaryl or propulsor. But if the infestation rate is high or a moderate and large number of animals are affected, then for mass treatment you can use a dipping method. So the dips can be constructed, or even I have seen the tubs can be used for individual dipping. The advantage with this method is that there is a uniform application. Okay, the entire body becomes wet because the animal takes in a dip and then it comes out. But as you go on passing the number of animals, the concentration of the acaricide or insecticide goes on reducing. So a concentration gradient is formed. And that is why, depending upon the size of the dip, you can pass say around 50 animals or 75 animals. And after that, the dipping solution has to be changed because the concentration goes on reducing. But in dipping, there are chances of toxicity. There are certain prerequisites for dipping. You should provide adequate drinking water for the animals so that they don't drink the insecticide solution. So the care has to be taken. Spraying is comparatively better, and for treatment of a smaller group, spraying can be used at the same concentration. And I came across one particular reference giving a systematic plan for spraying the body. According to that particular reference, you should start spraying on the back line first, then followed by on either sides and the flank region. Then you come down to legs, outer portion of the legs and inner portion of the leg. Then the underside of the body, thoracic region and abdominal region, maybe up to scrotum or the other portion. So the underbelly is treated. And at the end, tail and head should be treated. So a definite scheme has to be followed so as to ensure that there is a uniform application. And what I feel that, if you treat the head first and if you then go on treating the other portion, then the chances of toxicity is more. So head at the end, and then, the animals are let loose so that, you can monitor also properly. Then in case of systemic, there are tablets available, injectables are there, pour-on methods are there. So pour-on methods are very, what should I say, user-friendly method, very easy to, but there may not be equal distribution. Sometimes there is a question of bioavailability of the compound at different portions in the body. So these are different methods one can use. Now this chart shows the reports of resistance to different acaricides. And basically, you will find organophosphorus compound and pyrethroids. And this is mainly by Rhipicephalus microplus or Buphyllus microplus, which is a very, very common tick in cattle. Now, some of the tick, they are common between large ruminants and small ruminants. In large ruminants, there are number of references. There is a list of state-wise. So acaricide resistance or insecticide resistance is a big problem, and more the reason you should use these acaricides very, very sparingly. So, when we started, this ectoparasites is not new. Since last so many centuries, they are existing. So we, as a professionals, we started with vengeance, to eradicate them, to destroy them completely. But, we could not achieve this. So then we concentrated on controlling these ectoparasites. Even we could not control effectively. So we are now, targeting, or we are trying to manage these. Probably, if we manage these ectoparasitic infestations nicely, we will be able to control them. Certain common mistakes using pyrethroids. So what are the common mistakes? First is you are reconstituting the working solution under sun, because you will realize that these pyrethroids or even amitras are available in the amber color bottles or a metallic bottle, so as to protect them from the sun rays. And if you are reconstituting in the open sun, then there is a possibility of detoxification, or the potency may come down. So always reconstitute it under the shade. Once you reconstitute it, the dilution, that is 75 ppm or 100 ppm, you have to use it immediately. You can't keep the solution for use for the next day, because it is rapidly detoxified. It is of no use. You have to use entire reconstituted solution within next 2 to 4 hours. Then sometimes, the well water is used, the hard water is used, then the potency goes down. You have to use tap water, for reconstitution. Now, there are a few references stating that if you use a lukewarm water, when the temperature of water is, say, around 40 to 45 degrees centigrade, and then you mix the pyrethroids, then the potency is good, or the efficacy is also good. So these are some of the tips which you can practice in your day-to-day treatment protocols, and you will get a better results. In case of, mechanical removal, deticking, when you use it, you remove the ticks, and if you throw it on the ground, the ticks, without destroying them, then those ticks, are going to lay eggs. And then with one, tick throwing down, the level of environmental contamination within next 1 month. The level of environmental contamination would be around 10 lakhs of larvae if you just throw one tick on the soil. So the ticks should be destroyed, squashed, or you can put it in the kerosene, or you can put it in the liquid of pyrethroid and then destroy them. So these are some of the common tips. If you use it, you will be able to control the ectoparasitic infestations more efficiently. When you are using these acaricides or insecticides, when you use a particular compound for treatment of ticks and mange, it is known as acaricide. When you use it for insect, it is known as insecticide. When you are using it for lice, the topical application is really good. It is very efficacious. You don't have to go for systemic, because as I've said, these lice, they feed on superficial epithelial cells, biting lice or a dandruff. So if you use a systemically, the drug may not reach the ectoparasite. Okay, or even the sucking lice also, they take superficial lymph. So the concentration of the drug in the lymph may be very less. Okay, and you may not get the desired effect. So for treatment of lice, it is always better to use a topical application. While in case of mange, it is exactly a reverse position. You can use it particularly for sarcoptic and demolytic mange. Systemic use of acaricide is recommended, because these are burrowing mites. While for Psoroptic, yes, even if you apply locally, it will have some effects. But for a burrowing mites, it has to be a systemically used acaricide. Then for control of lice, shearing, if you do it and then you treat it, then there is an effective control, because with shearing 80% of the population can be eliminated. There are some situations where long wool is also treated, but then the number of applications are more. Then the remaining points are same, that cleanliness has to be maintained, disinfections of the farm application for both these cases. The only difference in case of mange, number of treatments are required, and the assessment of the clinical, the treatment should be there by two ways. One is a clinical examination. You can see that the lesions are healing up, and second is keep scraping and examine for the presence of mites. Resistance in case of mange is not reported, although for ticks the resistance has been reported, but for mange the resistance has not been reported. And then for mange, the supportive therapy is very important. For example, if it is a sarcoptic mange, you have to give some anti-allergic medicines. Even in extensive cases, you can use steroids. Then if there is a secondary bacterial invasion, then you can use antibiotics. You can use antihistaminics. So supportive therapy, you can use minerals and vitamins for a proper healing of the skin. So supportive therapy is required in case of mange. Then a number of treatments are required. While in case of lice, the number of treatments are comparatively less. Supportive therapy is not required, and the other aspects are same. These lice or mites, they can live away from the body of the animal for not more than 2 to 3 weeks. Then cleanliness, disinfection is very important. Desiccation is lethal for both these groups of ectoparasites. And if you apply all these steps, you can also cover, or you can also control Melophagus ovinus, which is known as a sheep cat. In case of flies, there are two groups as a myiasis producing flies and non-myiasis flies. In myiasis producing flies, there are like blowfly myiasis and screwworm fly myiasis. Blowfly myiasis is a common condition in sheep, and these flies are a facultative parasite. That means they don't require host all the time. Even if there is a dead decomposing organic matter, the life cycle can be completed. And whenever the animals are available having wounds on the body, then these flies then prefer these wounds to deposit eggs, and then the maggoted wounds are created. Then screwworm fly myiasis is also common in goats. It is obligatory. That means, these flies require necessarily host for completion of life cycle. So in both these cases, there is a cutaneous myiasis, and when the damage is extensive, even the subcutaneous tissues are also involved, and that is how they cause, loss of meat or a downgrading of meat and economic losses. In case of sheep suffering from blowfly myiasis, you will find that the body coat is rough, the wool is disturbed and discolored. So the careful examination reveals the maggots in the wound. Now, whenever you come across a case of a maggoted wound, field veterinarians commit an error there. They use insecticide in a hurry. Actually, the maggots should be removed in a, what should I say, live maggots should be removed by using a turpentine oil or a chloroform. You can just put a chloroform swab on the wound, and all those deeply seated maggots then come out because of the chloroform, and then you can, with the help of forceps, you can remove them. When you remove them one by one and clean the wound heals up very fast. If you use acaricide hastily or hurriedly, then these maggots die inside the wound. Then the wound is then the toxic excretion, they disintegrate, toxic excretions and secretions come out, and there may be irritation in the wound may be extended, and these wounds do not heal quickly. So ideally, the maggot should be removed from the wounds, and then you can use acaricide to prevent further infection. So dressing of such wounds then with the insecticide. Nowadays you get insecticide cream containing BHC. Okay, so that can be used. Fly repellents can be used. And then Estrus ovis, very common in India. As I've said, we have a slaughterhouse in Mumbai, and when we visit, their skull is divided into two portions. And in that particular room, I have seen hundreds of larval stages of Estrus ovis which are around 2 to 3 centimeters in length, migrating on the floor, hundreds of those larval stages. So that suggests that it is a very, very common infestation. And this is a fly called as a sheep nasal fly. This is a larviparous fly which deposit larvae on the nostrils. And these larvae then enter into the nasal passage and develop inside the nasal passage. Sometimes they enter the associated sinuses. Frontal sinus is commonly affected, and then they cause sinusitis. Occasionally, they may reach the brain and cause some nervous signs. So the nervous signs are exhibited, and that is why this condition is also known as false GID. As you all know, that GID is caused by Sinurus cerebralis, while this is a false GID, which is caused by Istosovis larvae entering into the brain. So when these are flies, they are hovering around the host in order to deposit the larvae on the nostrils. So in that, the animals get scared. They try to run away from the flies, and this is known as entomophobia. And while doing so, they may fall down. They may meet with an accident. So these chances are there. The sheep, in order to prevent the deposition of the larvae on their nostrils, they come together, with their heads in the center. I think you all must have seen in front of any game, the football match, all the players, they come together, and they make a circle. So a similar kind of approach is seen here, and keeping their heads very close to the soil so that these flies cannot deposit larval stages. So when the larvae are migrating in the nasal passage, they are responsible for causing rhinitis. There is a secondary bacterial infection. So mucopurulent discharge is there. Nervous signs may be seen occasionally. You can clinically distinguish between GID and false GID. In false GID, there will be nasal discharge. In GID, there won't be nasal discharge. So clinically, you can easily differentiate GID from false GID. And then as you can see here, this is the longitudinally cut skull, and where the larval stages can be seen in the sinuses. Now here, if you look at the life cycle, it is an annual life cycle. These mature flies, they survive hardly for 3 to 4 weeks. But the development inside the nostrils or even into the skull requires around 7 to 8 months. Then the pupal stages require around 1 to 2 months. So you will always find these flies in India. They are widely prevalent during summer months. Occurrence is annual occurrence. So just before the occurrence of these flies, you can take certain measures. You can implement certain measures like intranasal drops of sprays can be applied. Then rifaximin is a very good drug. Rifaximin, as you all know, used for treatment of fasciolosis. Rifaximin is also very good for the treatment of Haemoncus worms, and rifaximin is also good for the treatment of nasal bots. So rifaximin used in sheep and goat is really good. It is a wide spectrum endoectoparasiticide. Organophosphorus compounds can also be added in the feed. So that is how these infections can be controlled. So ectoparasites are very, very common in India. We have seen prevalence, and that is the reason they are associated with enormous economic losses. Unfortunately, in countries like India, it has not been estimated, but I am pretty sure, I have no doubt whatsoever in my mind that the losses associated with ectoparasitic infestation must be pretty high. Huge losses are there, and the poor farmers, they wouldn't appreciate, they don't know the losses associated with these ectoparasites. Because of these ignorance, they don't take these ectoparasitic infestations very seriously. There is a job of a veterinarian to educate them, to tell them the significance of these ectoparasitic infestations. And that is why my suggestion, my recommendation is that when you come across the ectoparasitic infestation, tackle it at the first instance. This is the point where the treatment is very easy. If you allow these ectoparasites to multiply in the particular flock, then the treatment becomes very, very tricky and tedious. as far as possible, your treatment should be eco-friendly. So try to develop a IPM module, like, deworming schedule. There is no uniform deworming schedule. Likewise, you have to develop IPM module in your area, in your region, to treat it and minimize the use of these chemical ectoparasiticides. And that is how you can control these different ectoparasites.